Hello students, the topic that we'll deal here is callus culture. The callus is an undifferentiated, unorganized mass of proliferating cells, generally parenchymatous in nature. In general, a plant produces callus at the region of wound or regions of infection due to production of many endogenous growth hormones like auxins and cytokinins. A plant tissue culturist can manipulate the pathway taken for organogenesis or embryogenesis by varying the concentration of auxins and cytokines. In general, for callus production, the explants are inoculated in a media containing equal concentrations of auxins and cytokinins. Later, by increasing the concentration of auxins to cytokinins leads to root production. Further, increase in ratio of cytokinins to auxins leads to the production of shoots. This is known as indirect organogenesis. As the organogenesis happens, that the shoot and root formation happens after the formation of an intermediate stage, that is the callus. The process of callus culture includes excision of explant from the mother or the donor plant, followed by surface sterilization of the explant and inoculation into a suitable nutrient media. This is then incubated in culture rooms, and the maintenance of rapid proliferation stage is done by subculturing at frequent intervals. The whole process can be divided into three stages. Stage 1 includes the induction or initiation of proliferation. This stage includes the excision of explant, surface sterilization of the explant, followed by inoculation of the cells into the media. Stage 2, the proliferation stage. In this, the plant explant is maintained in plant growth regulators to induce rapid cell division and make a mass of calli. Stage 3 is called morphogenesis, which includes change in the concentrations of the phytohormones so as the undifferentiated mass of cells get differentiated into organized structures of roots or shoots. This is with an intention of for plant regeneration. The callus varies in texture and coloration based upon the different hormone concentrations that are used. A callus could be soft or hard. A soft callus is friable in nature and is made of heterogeneous mass of cells having minimal contact. This is the callus that is actually preferred for suspension culture. The figure at the bottom last shows the soft friable callus. Hard callus are nodular in form. The callus consists of giant cells, trachete-like cells, and closely packed. Figure A, the first one, shows a hard, compact green callus. The color of the callus may also vary from creamish yellow, white to pigmented or green. The flowchart here summarizes the different ways a callus can be utilized in plant tissue culture. The explant, when inoculated in the induction media, induces cell division, dedifferentiation, and high proliferation to form undifferentiated mass of cells known as calli. Depending on the type of callus, these callus can be utilized as protoplast donor cells for transformation, somatic embryogenesis, organogenesis for cell suspension culture, and secondary metabolite production. Plant regeneration from callus culture can occur through organogenesis or somatic embryogenesis. Organogenesis is the formation of individual organs such as shoots or roots, while somatic embryogenesis is the formation of bipolar structures which have both root and shoot meristem, and development happens similar to that of a zygotic embryo. Now, organogenesis in plant tissue culture can be direct or indirect. Direct regeneration means regeneration of organs, that is shoot or root, directly from the explant. The figure on your extreme left shows regeneration of shoots from a leaf explant. Indirect regeneration means regeneration of shoots, roots, or even somatic embryos from the callus. That is, the explant is first induced to form the callus, and then by manipulating the growth hormones, the callus is induced to form shoot, root, or both. The figure on your extreme right shows indirect regeneration. Applications of callus culture. The callus culture can be induced for organogenesis or embryogenesis by manipulating the growth hormone concentration. Hence, one can get large number of plantlets from the callus. Now, callus can be a source of genetic variability, leading to the formation of somoclonal variants. A callus can be utilized for commercial production of secondary metabolites. Large number of biochemical pathways can be manipulated in callus culture to get high amount of secondary metabolites. The main limitation of callus culture is it may lead to chimeras. Thank you.